Okay, welcome everybody. Thank you very much for attending. So it's my great pleasure to welcome Dr. Blair Leavitt to the CBR, who joined us from very, very far away, Oak Street. So Dr. Leavitt is a neurologist and a clinician scientist and a full professor in the Department of Medical Genetics. His lab is at the CMMT at BC Children's Hospital. And Dr. Leavitt received his medical training at McGill University in Montreal, and subsequently his neurology and research training at Columbia University, Cornell University, and Harvard University. So it's some fairly well-known institutions there. So Blair is the director of research at the UBC Center for Huntington's Disease, and we're very happy to have him here. Incidentally, I actually didn't meet Blair through UBC. I actually met him on the hockey rink. And so when I joined this hockey group a couple of years ago, the first few months that I was playing, anytime I was on the ice against him, my shifts invariably ended with me digging the puck out of our net. So, but nevertheless, we're very happy to have him here. Blair, thank you very much for speaking today.
Thank you very much, Hugh, for the kind invitation to talk today. I really appreciate it, and congratulations on your new position. Well deserved. I think you were exaggerating a little bit about the hockey stuff, but you were pretty accurate on the rest of the CV. Okay, so my lab at the Center for Molecular Medicine and Therapeutics has really worked in therapeutic development for genetic brain disease, and primarily the approach that we use is we develop a mouse model and then we try and fix the mouse model. We work in a number of different disorders: frontal temporal dementia caused by progranulin mutations. We work in pyridoxine-dependent epilepsy and a number of other genetic diseases that affect the brain. We've worked in ALS, but most of my work has been focused on Huntington's disease. So it's a little bit of a misnomer here. I'm only going to talk about one disorder. I'm only going to talk about Huntington's disease, tell you a little bit about the disease, and then how a crazy idea that we had in the lab has now led to patents, a company, and ultimately leading into a therapeutic that will be going into the clinic. I do have some disclosures because I'm actively involved as an advisor for a number of companies developing therapeutics for Huntington's disease. I get research support from a number of sources, do other things, and importantly, I am actively involved. I'm currently on a 50% leave of absence from the university, so half of my time is at the university as a professor, and half of the time is the CEO of Incisive Genetics, a BC-based biotech company that I'll tell you about. Because I'm going to be talking about it, I've got to put up something like this. No one ever reads it, but don't make any investments based on anything I say today. I think that's what that means. So I'm going to tell you a little bit about Huntington's disease. I'm not going to dwell on it, but just so we're all on the same page. I'm going to tell you about some of the current therapeutics that are in development and our role in some of that. Then I'm going to introduce you to the delivery system that led to Incisive Genetics and the crazy idea that actually in the end seems to have panned out a little bit. And then we should have lots of time at the end for questions. So happy to answer any questions you may have. So I've spent most of my career, certainly all my clinical time, focusing on Huntington's disease. This is a devastating autosomal dominant disorder. And almost every paper I've had to write has to include a little phrase, something like this, that it's a hereditary neurodegenerative disorder, and there's currently no known treatment that alters the progression of this disorder to complete disability and death. And unfortunately, despite 25 years of work, I haven't made a dent in that yet. It's an adult onset disorder for the most part. It's very slowly progressive, affects specific brain regions. And that's about all I'm going to tell you about Huntington's disease, other than a little bit more about the genetics and the pathophysiology. We won't be focused on the clinical aspects today. So what causes Huntington's disease? I imagine most people in the room know. This is caused by a single dominant disorder. So everyone who has this disease has the same underlying genetic mutation, which is a CAG trinucleotide repeat expansion within exon 1 of the Huntington gene, abbreviated HTT. And we know that this exonic expansion of CAGs, which are encoded into a polyglutamine within the protein, affect the function and induce a toxic gain of function of the mutant Huntingtin protein. And because of that, people have studied extensively how mutant Huntingtin affects cells, specifically how it affects brain cells. And basically, if you have a favorite mechanism of toxicity, throw mutant Huntingtin at it and you'll probably find some abnormalities. It mucks up pretty much everything in the cell, mitochondria, proteolysis. It has transcriptional dysregulation when it gets into the nucleus. A lot of really exciting work from UBC, led primarily by Lynn Raymond, has shown that excitotoxicity may play a really important role in the regions of the brain that are affected by Huntington's disease. It can aggregate, much like alpha-synuclein or A-beta or other toxic proteins that cause neurodegenerative diseases. It affects neurotrophin basically, it does pretty much everything. And because of this, people have really focused on mutant Huntingtin and the protein and how it causes disease. And some of that may be actually the fault of those of us that make things like models. Because how did we make Huntington's disease in a mouse model? We express very high levels of this protein with very large expansions to try and accelerate the process so that we have a phenotype that we can measure. Because of this, there's a very simple hypothesis. All you need to do is get rid of this protein and you're going to treat the disease. It was a really brilliant hypothesis. We thought it was going to work great. And there was a huge number of therapeutic modalities that were directed at specifically, let's get rid of Huntingtin, lower mutant Huntingtin, and we will cure this disorder. And there are many different approaches that are taken. We can target downstream from the protein. So there's lots of protein clearance mechanisms that might be affected or protein stability. But from a genetic point of view, we can target the mRNA with small interfering RNAs. We can target the pre-mRNA with antisense oligonucleotides. Splicing has been something that's been targeted, and there are actually splice modifiers that are now in the clinic that decrease the amount of the Huntingtin protein that's produced. And then we can also target the DNA. And I'll tell you a little bit about our approach using CRISPR-based editing in a bit. But before that, I want to take you through one of the approaches that we were involved with, bringing all the way to the clinic, and that's actually still in clinical development. That's using an antisense oligonucleotide approach to decrease and lower Huntingtin levels in the brain. So this was an approach that was developed originally by Ionis, and then subsequently brought to the clinic and is still in the clinic being taken forward by Roche. Antisense oligonucleotides are short artificial strands of DNA that are antisense to a target. In this case, the target is the Huntingtin pre-mRNA. So they bind to that pre-mRNA and they form a DNA-RNA hybrid, which in the case of the Huntingtin ASO is recognized by an enzyme RNase H and degraded. You degrade the mRNA, you prevent it from being translated, and you decrease the amount of protein. Pretty straightforward. Antisense oligonucleotides have a number of really attractive qualities as a drug development tool. If you get them into tissue, they diffuse pretty freely. They have a dose-dependent effect, and that dose-dependent effect is reversible, so it's not a permanent treatment, and that has some advantages. They're relatively stable when they're taken up within a cell, and so you can have very long-term action. And most of the ASOs in development have durations of actions in months, not days or hours. So long-acting, reversible, but with a good penetration. Unfortunately, they do not cross the blood-brain barrier. And so in order to deliver these to patients, we had to move to a system that actually really hadn't been used previously in Huntington's disease. And this was to deliver directly into the cerebral spinal fluid, bypassing the blood-brain barrier. And it's basically just the reverse of a spinal tap. In fact, it was a spinal tap. We took off about 20 cc's of spinal fluid, and then through a bolus push, delivered antisense oligonucleotides to patients. This had never been done in Huntington's disease, but there was precedent in other disorders. There was an ASO developed by Ionis that was showing promise at the time in spinal muscular atrophy, and which subsequently has been approved as a therapy for that disorder. So at the time, we were working very closely with Ionis. The drug was called RG6042. Eventually, actually, it was originally called Ionis HTTRX. Then it was RG6042, and eventually it became known by the name it's currently called tominersen. And this is the first actual genetic therapy trial in Huntington's disease. And this study came out of about 15 years of work. So in 2005, they first showed that you could target Huntington with ASOs in cell models. Some really important work by Korashevitz et al. Showed that in various Huntington's disease mouse models, you could target Huntington. And at that time, they didn't have the ability to selectively target just the mutant allele. So they developed very efficient and very effective ASOs that target all Huntington. So as I mentioned, this is not a somal dominant disorder. So everyone has one normal gene and one abnormal gene, and the abnormal gene is what's producing the toxic protein. But these therapies target all Huntington. And we'll talk a little bit about that probably in a minute. So this first work in mouse models, and they actually used in most of the studies, one of the mouse models that was developed here in my postdoc with Michael Hayden, the Yak 128 mouse model. And they also showed that if you target mouse Huntington at the same time you target the human transgene, you still got benefits. So there was a belief then that we could do total Huntington lowering and it would be beneficial. At around the same time, our group and some collaborators from the University College London had developed a technique for identifying mutant Huntington within cerebral spinal fluid. So cerebral spinal fluid does contain a variety of proteins, but at very low levels. So we had to identify some ultra-sensitive detection methods that allowed us to detect mutant Huntington. This is critical because that's a biomarker that we can use to measure the effect of our therapy in a person. We can't take out the brain like we do in a mouse or a monkey and measure the amount of Huntington lowering. We have to do it indirectly through a biomarker. And the biomarker that was developed actually in parallel with the ASO was mutant Huntington detection in the CSF. So this all sort of came together in about 2015. And actually Vancouver was the site of the first ASO infusion. This is a very happy neurologist who has just infused the first patient with Huntington lowering therapy in September of 2015. Everything was groovy. 46 patients were recruited into the study, multiple ascending dose study. So we gave the lowest dose first, seemed safe and well tolerated. We gave a subsequent dose to the next group, made sure it was safe, and eventually got up to 90 and 120 milligram doses. This was done monthly for about 4 months. And what we saw was a dose-dependent effect after 4 months on Huntington levels in the CSF. Beautiful. And if we look at the two highest dose, you can see 4 months of repeated dosing, we get a nice, consistent lowering of mutant Huntington in the CSF. Couple things to note. This is fairly frequent dosing. I mentioned that these ASOs have a long half-life. Turns out much longer in brain than we had actually realized. And we've done some postmortem analysis on an individual from this study who did eventually pass away through the MAID process. And turns out that we were nowhere near the bottom of this trough, but it did what it said. All 46 patients completed the study. We had a beautiful dose-dependent effect, and New England Journal accepted it. Everyone's excited. We've cured Huntington's disease. We've lowered total Huntington. Roche bought the program from Ionis. The original phase 1/2A trial of 46 individuals was rolled over to a long-term open label extension. So now everyone was on the drug for an extended period of time. They matched that with a natural history study. So we had someone to compare. So for every patient that was on the open label, we had individuals who weren't being treated that we followed, and then went straight into a huge 800-person phase 3 definitive trial with a maximum dose given every month. We had some early data to suggest that that was too frequent. So the dosing interval was changed, but the dose wasn't. And then unfortunately, about 6 years after everything started in 2021, height of the pandemic, everything was stopped. The Data Safety Monitoring Board recognized that not only was this not having benefit, but the individuals in the highest, most frequent dose were actually doing worse. pretty devastating outcome. Everything was stopped, and there was some hints. This is actually from one of the early meetings we had with Roche, where we were looking at some of the findings from that first trial, and there was evidence of some elevations of neurofilament with this frequent dosing regimen. And there was also evidence that the ventricular size was getting larger, and it was a dose-dependent effect. So probably something that shouldn't have been ignored, both the neurofilament and the changes in imaging. But unfortunately, and actually, despite some of the recommendations of their advisors, they still went forward in the phase 3 with the highest dose, really informed by other disorders like Parkinson's, where nothing had ever done anything. So they wanted to go with the maximum dose. Turned out to be a bad idea. It is now still in clinical trials, in phase 2 trials, so they backed off, and they're looking at a couple of lower doses. We don't know the outcome of that. Those studies are still ongoing. But I think we need to learn from this. And one of the things we need to learn is that probably simply targeting all Huntington was not a great idea. We know that normal Huntington, wild-type Huntington, has some really significant roles. It's certainly, everyone knew it had roles in development, but we've done a lot of work in the past to look at what the role it may have in the adult brain. And it basically has a whole series of neuroprotective effects. If you overexpress it, you protect the brain from stroke, from traumatic brain injury, from a whole series of insults. And if you take it away from the adult brain, you make the brain more sensitive to insults. Now, there may be a window of opportunity where if we lower total Huntington, we take away the mutant, and we still have enough wild type that you may see benefit, but it's not clear that that will ever be the case. Ultimately, we'll know from these Roche studies. But since 2015, there's also been a lot of new elements of the Huntington's disease pathogenesis that have been identified. And what's really important, it's been just really a hot topic in the last 5 years, is somatic instability. This was actually first discovered in '93, right after the gene was discovered. Dr. Michael Hayden identified that brain regions that were most affected by Huntington's disease had this weird change in the CAG repeat size, in that it got bigger within regions of the brain that had most toxicity. And this was kind of known, but then forgotten for many years until large GWAS studies were done, and they identified mismatched repair enzymes, or DNA repair enzymes, as being modifiers of the age of onset. And no one could really put that together until it was recognized that these influenced this somatic instability. And there have been some really elegant studies from 2 large groups, Steve McCarroll and another group in New York, which I'm forgetting the name of, I apologize, looking at single cell analysis. And when you look at a single cell analysis where you can actually measure the CAG repeat size and transcriptional dysregulation in the same cells, it's really clear that as this somatic instability occurs, you get a dramatic peak in transcriptional dysregulation and presumably toxicity above a certain size. So it's clear that simply the expression of the protein isn't all. In addition, these very large repeats induce a splice variant called HTT1A. So you don't need to express the whole protein. You can actually get a splice variant that comes off just off exon 1, and it produces a very small fragment that is almost exclusively polyglutamine, highly toxic, and it occurs at high levels when you get these large expansions. So this is all activity that's going on at the DNA and producing splice variants and peptides, whether through cleavage or through this splice variant, that have nothing to do with the ultimate product of the gene, which is the protein. In addition, I mentioned we think that wild type Huntington function is important, and we probably want to spare that as much as possible. So there are also some RNA toxicity mechanisms that may be involved where the protein may not be relevant at all. So we've learned a lot since that simple, if we knock down Huntington, we're going to cure this disease. I'm not saying that Huntington lowering is a dead therapy, but I think we know a lot more now. So if we look at what the current landscape is, the only thing approved in patients are symptomatic therapies. These tetrabenazine, deuterobenazine, these are just medicines that control chorea. There is one drug that is still being evaluated by the European Medical EMEA. And that's predopamine. It's a drug that may have some modest neuroprotective effects. It's being evaluated in ALS as well as Huntington's disease and may yet show some benefit. I mention it partly because Prolennia is a company started by Michael Hayden. He took this drug from Teva, where he was working, and is keeping it going as a potential therapy for Huntington's disease. If we look at the drugs in development, PTC has a splice modifier, and tominersen, the ASO I told you about, are both total Huntington lowering drugs. They're in phase 2. Maybe if we get the dose right, there is a window of opportunity where we can see some benefit. But I think they're unlikely to be very effective or long-term cures for this disorder. There are a number of approaches in earlier phase. Unicure has a small interfering RNA delivered by an AAV. Voyager is trying to develop a similar approach. Wave has an ASO that may be allele specific. They've had 2 that have failed because they don't target Huntington sufficiently. Vico is another ASO. This is directed towards the CAG repeat. And Alnylam has a modified siRNA that penetrates the blood-brain barrier. All of these, again, are targeting Huntington, whether it's allele specific or not, and they're all targeting downstream at the RNA. There are a few other approaches in development, but basically what's clear is that as we've learned more about the pathogenesis of this disorder and the role of the upstream DNA changes that lead to somatic instability and the exon 1 fragment, none of these are going to target all of those mechanisms, except one, which I'm going to tell you about, which is IGHT01, the product that is developed by the company that we spun out. So before I get into that, I'm going to tell you a little bit about where it came from. Where did our Huntington's disease therapeutic come from? We've been working in, as I said, various forms of genetic delivery. We've worked with AAVs, worked extensively with Beth Simpson and others, looking at AAV delivery to the brain. We've done ASO work, as you've seen, with Ionis. Alnylam has developed modified siRNAs that penetrate the blood, penetrate the brain efficiently. But it was really some work with lipid nanoparticles that got us very enthusiastic about a novel way of delivering gene therapy and genetic therapies to the brain. So lipid nanoparticles are complex delivery systems. They are composed of a variety of different lipids, phospholipids, but primarily cholesterol with these other lipids. And they are able to very effectively encapsulate nucleic acids. And this is well known. In fact, probably everyone in the room has had a jab with an mRNA vaccine that is LNP mediated. But really, and they're very effective. If you encapsulate something within a lipid nanoparticle and you get it to a cell, the lipid nanoparticle takes up lipoproteins and basically co-opts the cell's cholesterol delivery system. So you go to the doctor, you get your LDL measured. Well, what you're really measuring is the lipoproteins in your blood. Those lipoproteins are taken up by cells, taking cholesterol into the cells through receptors. And specifically, the LNPs that we use are taken up by the LDL receptor. They're in endocytosed and then released from the endosome into the cytoplasm where they can act. And we got very enthusiastic about this, really from pioneering work from Peter Cullis and Brian McVicker's labs here at UBC. Peter's been a sort of the godfather of lipid nanoparticles, and we worked with him for many years. But it was really the ability, if you deliver these to neurons take them up very efficiently. And we got very enthusiastic about this. And we did some work early on with siRNAs and with mRNAs, and sure enough, beautiful. Apply them, neurons, whether it's in vivo or ex vivo, are very hard to transfect. We were seeing levels of transfection with very little toxicity that were miles beyond anything we'd ever used. And so this is what got us excited. Hey, we've got something we can now genetically manipulate neurons. And, we did it in neurons from our Huntington's mice, and we're able to show very efficient knockdown of Huntington. That was great. We worked with Leslie Thompson's group in UC Davis. She had human iPSC-derived neurons, and in those cells, again, very efficient knockdown of Huntington using siRNAs. And in mice, we were able to just put it into, just basically inject it into the brain. And sure enough, if you get it to the brain, to neurons ex vivo or in vivo, they take up these LNPs very efficiently and knock down transiently Huntington. So this is where we were. We had a couple of small grants with Peter. We actually tried to see whether encapsulating ASOs within lipid nanoparticles would be more efficient, but ASOs were taken up so effectively it didn't really matter. But we could deliver a number of cargos, nucleic acids specifically. We were able to reduce the Huntington gene using siRNA, worked both ex vivo and in vivo. LNPs actually have some real significant advantages. They don't induce an immune response, so you can do repeat dosing, and they're already proven well to be safe for in vivo administration. At around the same time, we were starting to do a lot of work with genome editing and CRISPR, as probably everyone in the room has. It's changed the way we do science, and I think probably people understand it. But I'm just going to take you through quickly what the very bare minimum of what's needed to understand what CRISPR does. So you really need a minimum of two components. You need your enzyme or your endonuclease that does something to the DNA, and you need an RNA, a guide RNA, that will target that effector protein to the DNA. They have to come together very specifically to form a ribonucleoprotein complex, or RNP. And it's the RNP that is the actual effector. And that's a relatively complex thing to try and deliver. The RNP then can bind to the DNA, and depending on what effector you use, it can either cleave the DNA, it can create nicks, it can methylate or demethylate, you name it. As you know, the world is full of CRISPR-based approaches, but all of them require those two steps. They require a guide that directs it to the DNA and a protein that does the effect, and they need to form the RNP complex. Sometimes you may also need a DNA template. So now we've got three different components: a protein, a guide RNA, and a DNA. Doesn't sound that complex, but to deliver all of those things into a cell efficiently and safely so they act together is the problem. And as I think everyone probably knows, Jennifer Doudna was one of the Nobel laureates. And in 2020, she really gave a beautiful lecture at World CRISPR Day, where she identified that delivery is really the gateway to that. If you can get these effectors into cells, they work beautifully. The question is, how do we deliver it to the body, to tissues where it's required for it to be useful? And that's really the gateway. And there are a number of challenges to what was being used or what's still currently being used. The primary approach is to use viruses. Viruses can only deliver nucleic acids. So now you need to deliver an mRNA for the protein, a guide RNA. It has to be delivered either at the same time or has to be produced simultaneously. That's very inefficient in the cell. If you use lipid nanoparticles, again, you're delivering the individual components. So they have to arrive simultaneously. They have to form the complex before they can act on the DNA. And if you're producing it with a virus, producing unbound Cas9 or any endonuclease for extended periods of time is a very bad idea. People may try and convince you it's not. It is a bad idea. You don't want endonucleases floating around in your cell, unbound to a guide, doing whatever they want. It's a bad idea. So in parallel, we were working with Beth Simpson. She was making mice using CRISPR-based transgenesis, and she was actually the first one to tell us that, no one would deliver the individual components if you can deliver the RNP. And she could do it because she was doing pronuclear injection to create animal models. And so we said, well, if that's the gold standard, if everyone knows that, well, why aren't we trying to do that? And well, you can't do that with viruses, and you can't do that with LNPs. You can only deliver nucleic acids. And we went to Peter and we said, Peter, we think this is a great idea. We want to, deliver RNP with lipid nanoparticles. And he just kind of laughed and said, silly neurologist. You're not a chemist, you don't know what you're talking about. And he was right, because for about 5 or 6 months, we tried and didn't work. And then it did. The only approved therapy, or sorry, I should say, the first approved, because this changes all the time, the first approved CRISPR-based therapy actually uses delivery of the RNP. It's Casgevy for sickle cell anemia. In that case, you're basically doing a bone marrow transplant. You take the hematopoietic stem cells out of the body, use electroporation, or basically electroshock, to deliver the RNP into the cells, and then you transplant them back. So we thought that this was, now in 2025, validation of our idea back in 2018, that if we can deliver the RNP with LNPs, that's going to be highly effective. And there's a large number of advantages. And actually, I think the field is coming around. Even David Liu, well known in the CRISPR field, has come out saying that ultimately we need to deliver RNP. And we identified a method, changed the process by which the LNPs encapsulated their cargo, that allows us to encapsulate the preformed RNP complex. This has some real significant advantages. The RNP is ready to act. It acts on the DNA, whatever you want it to do, cleave, nick, methylate, demethylate, block, whatever. It acts, and then it's rapidly cleared. There's no in-cell production of unbound Cas9. You can use much lower doses. It's highly efficient, has a much better safety profile. There's lots of data showing that it has less off-target effects, and it has very high rates of gene correction, because we can also include within that lipid nanoparticle DNA templates. So now we're delivering everything in a single shot. We can include multiple copies of the guide RNA, so we can target multiple genes or even multiple regions within a gene. So it was this really crazy simple idea that we can take lipid nanoparticles, which we were really enthusiastic about, and encapsulate a different kind of cargo, the right cargo for genome editing, the preformed RNP complex. We brought this to the UILO, we patented it, and eventually it led to the development of a company called Incisive Genetics. It was founded in 2018 based on this idea that we can encapsulate preformed CRISPR RNP. So far, we've raised about 17 million. We have 18 employees, and we've built out purpose-built laboratory and office space in East Van. Most of the employees all have come from UBC. The two of the co-founders, Austin Hill and Pam, were in my lab for 15 years and are now leading Incisive Genetics. And we've built out a whole platform of technology. So this LNP, RNP delivery system can deliver to the brain. It can deliver to the bone marrow through direct injections. We can deliver to the eye. And if you give it systemically, most of it ends up going to the liver, so we can target liver as well. I think I've kind of mentioned the advantages. I don't want to go over it too much. It's starting to sound a bit like a sales pitch. But we've shown that it's not just Cas9. We can encapsulate really any that we've tried. We've tried Cas9, Cas12, base editors, prime editors, various proprietary enzymes from other companies. We can include DNA templates, and importantly, we can include multiple guides targeting either multiple regions in the gene or multiple genes. And that's really one of the advantages of these LNPs. They don't really have a cargo capacity similar to the limitations with viruses. And critically, again, we can do repeat dosing. This means that unlike viruses, where everyone in the world is trying to give the maximum titer that is safe and tolerated because you got one shot, you can't keep giving viruses, you develop an immune response. We can do titratable genome editing. If we get 10, 15% editing with a single shot, we've shown that repeat dosing allows us to get 30, 45, 50% editing. So we haven't done a lot of direct head-to-head comparison, but we did do one experiment early on. Very simple system, 293 cells, human immortalized kidney cells. We use this for a lot of our biochemistry and CMC work, so chemistry work with our various lipid nanoparticles. We have a very standard 50 nanomole of a Cas9 with a guide against Huntington, and it gives us very reliable 50% knockdown. And we said, well, let's just compare that 50 nanomolar dose with the equivalent if we now use mRNA. And we really couldn't get much editing, even though we're now doing this in a dividing cell type, an immortalized cell that's super easy to transfect. I can tell you that I have a graduate student who worked in my lab for 4 years trying to get mRNA CRISPR-based genome editing in neurons and in mouse brain, and we've never seen above 1 or 2% editing in those systems. mRNA is just not efficient enough. You can't deliver all the individual components for CRISPR-based editing and expect to get high levels of editing. So orders of magnitude 10 times more of the mRNA, the max we could basically get into a lipid nanoparticle, we still don't get the equivalent of a very low dose of RNP. This is just some of our preliminary data. We throw it on primary mouse neurons. These are neurons from a Huntington's disease mouse model. We get very effective knockdown. This is, again, some of our very early work where we're looking at total Huntington lowering. We could do it with iPSCs again. So similar to what we did with Leslie Thompson, we now are using CRISPR-based editing to target iPSC-derived neurons. Again, pretty efficient. We got hit by the pandemic, so we shipped some of our total Huntington lowering LNP, RNP to Charles River in Finland. They were able to show that, yes, stick it in the brain, you get Huntington knockdown. We were, at that time, because of some of the Roche data that I showed you, convinced that we had to be more specific and more selective. So we started working on allele-specific targeting, thinking that total Huntington lowering wouldn't be efficient. And we can get, if we target SNPs that are on the Huntington gene, we can target selectively one allele versus the other. And interestingly, because our RNP system is so efficient, we can actually introduce additional mismatches. So targeting a SNP that is only present on the mutant allele has one mismatch with the wild type. So it's less efficient binding to the wild type. But if we introduce two mismatches, now one mismatch on the mutant and two on the wild type, it's still efficient enough. We still get pretty good targeting in the mutant, and we get really no targeting on the wild type. So that actually, because of the efficiency of our system, we can modify our guides and make them less efficient, but more selective. So that's actually part of the underlying patent behind the approach that we're going to be using. We did some non-human primate studies here. We couldn't, the non-human primate don't have the human gene, so we were targeting monkey Huntington. We can deliver Cas9 to the caudate or the putamen in this case. We can get good distribution. We can get evidence of editing, but really, that wasn't the approach we wanted to move forward. And I'm only going to tell you about our Huntington's program, but I'll just mention now that we also have programs that we're working with a partner for in vivo bone marrow editing. So basically the same approach used for Casgevy ex vivo. We now use our LNPs to do direct delivery to the bone marrow. This is going to democratize this therapy, which so far about 100 people in the world have had. Maybe 7,000 people eventually will get that therapy. There are hundreds of thousands of people that need this therapy across mostly, sub-Saharan Africa and India, where bone marrow transplants are never going to happen, or I shouldn't say never, are not happening currently. We also have a program targeting the liver, but I'm going to focus on our Huntington's disease program. This is a program that went from this crazy idea of can we do LNP, RNP, we're now moving into the clinic. We're in the midst of a raise. It's expensive. It's going to cost about $50 million to take this all the way through our first in-man clinical trial. We're working with the Huntington Study Group to develop our clinical trial, but I'm going to tell you a little bit about our HD therapy. So this is IgHD01, and it uses our proprietary lipid nanoparticle delivery system, and it has Cas9, standard good old SP-Cas9, with two different guides. And again, all encapsulated within a single lipid nanoparticle. We can include a DNA template, and that actually increases the efficiency of the system. And we've targeted two places in the Huntington gene, upstream in the promoter and downstream at a SNP. The promoter is a region where we can introduce cuts, and it doesn't affect transcription, but it is highly efficient at introducing a double-stranded break. The SNP, as I said, we can very selectively target by introducing an additional mismatch where it will not target in our hands, and we've never seen targeting of the wild type allele, but will still, with some efficiency, target the mutant allele. This now leads to very large deletions, eliminating all of exon 1, the CAG repeat, and a number of downstream exons. So we're basically cleaving out in an allele-specific way all of the upstream DNA that leads to Huntington's disease. So as I said, it's a single mutation, but it has complex pathology. By deleting the region of the mutation, we're eliminating all of these pathologies. We're eliminating exon 1. You can't get an exon 1 splice variant if you don't have exon 1. We're eliminating the CAG repeat. You can't have instability if you don't have the repeat. It does eliminate mutant Huntington expression, and it prevents other forms of toxicity. And because it's allele specific, we're sparing the wild type, so we don't have loss of Huntington function. We think that this is an elegant solution to a critical problem. It takes into account all of the known disease-causing mechanisms. It targets as upstream as we can. It leads to correction at the DNA. Once that DNA is corrected in a cell, it's permanently excising the cause of the disease. So can we do this? Can we act? And we're actually, it's going to probably blow some people's mind. We're excising about 150 kb of DNA. Didn't seem like this would be very efficient. And when it was first proposed as an approach, I didn't think it would work. Shockingly, it's really highly efficient. And we can identify that chimeric, what we call HTT delta. So when you excise it, you now bring two regions that are very far apart in the genome together. And with a simple PCR analysis, you can identify that chimeric DNA. So we can see in human iPSC-derived neurons, very efficient delta HTT production. And importantly, when we look downstream, this is, again, those Huntington iPSC-derived neurons. When we look at the untreated group, you've got 100% of total Huntington, and it's 50-50 wild type and mutant. After targeting with IGHDL1, you get selective depletion of the mutant form with no change in the wild type. Highly selective excision of the pathogenic cause of Huntington's disease. So, oops, sorry, apologize. So because it's allele specific and it targets a SNP, it's not going to target everyone. You have to have the SNP. It has to be in phase with the mutant allele. Lots of work has been done in this. Michael Hayden's group has really led the world in identifying these SNPs and their frequency in various populations. The North American population, our first product will treat about 48% of patients with Huntington's disease. We have 2 follow-ups. All they vary is a slight difference in the sequence of one of the guides. They use the same promoter guide. Now it's slightly different sequence. This gets us up to close to 80%. That's a pretty big number, 80% of the population in HD. One of the things that is a real delay in going from, I'm a simple laboratory scientist, how do I bring something into the clinic is it's actually sometimes really hard to make things in what we call GLP or GMP quality. So to bring things to the clinic, you've got to be able to manufacture it. We started this before the pandemic. We've all seen how rapidly LNP therapies can be brought to the clinic. And the exact same method of manufacturing is what's used to encapsulate our product. And Danaher actually has the entire ecosystem from start to fill finish. So fill finish is what is used in the clinic, where they produce GMP quality protein, specifically Cas9 and Cas12 and others now. IDT, they purchase IDT, they make the guides in clinical grade quantity, and they purchased Precision Nanosystems, another one of Peter's companies, now called Cytiva. So they now have start to finish RNP, LNP encapsulation, ready to go into the clinic. This is very exciting, and they're actually now in negotiation with us to use our specific modification to the manufacturing to potentially to be the commercial producer for LNP, RNP. How are we going to deliver this? There's a system developed by Renishaw. It puts in catheters into the brain. The standard for viruses is you stick a needle in under MRI guidance, the patient's under anesthetic, you infuse slowly over 8 to 9 hours, and then you take the needle out. Great, but that's 8 to 9 hours under anesthesia. And if you wanted to do another dose, you have to stick another needle in. The Renishaw system puts in catheters. It takes about 30 minutes per patient, 4 catheters into the target regions. Patients come out of anesthesia after 30 minutes or an hour, and then you can slowly infuse them outside of the OR in an infusion suite. The catheters are permanent but can be removed at any time and allow us repeat dosing over time. This is a huge advantage to us where we can now start with low doses and titrate up as needed with repeat dosing. We're doing our large animal studies in sheep. There's some real advantages. First of all, the Renishaw system has been proven to work in sheep, and it's the exact same system used in people in sheep. We have a sheep model that actually contains the full, it's a cDNA, but fortunately for us, it actually contains the sequence that we're targeting. So we now have the human gene in a large animal that we can target. The sheep produce mutant huntingtin. It can be measured in the CSF. That biomarker I told you about is available in the sheep. So we can target the sheep with our human-based therapy using the same delivery system from Renishaw, and we can measure mutant huntingtin in the CSF. We can also measure neurofilament. That protein that I mentioned briefly goes up with toxicity in the brain. So hopefully we want to see mutant huntingtin go down, and while we might see transient increases in neurofilament, we also want to see that go down. And the same imaging, PET and MRI that's available in our human studies are available with the sheep. And this is sponsored by CHDI Foundation who developed the sheep. So we're working with the Huntington Study Group, and we've got a design for the first-in-man clinical trial. One of the real advantages of the approach that we're taking, I believe, is that by implanting these catheters, we can do a true placebo-controlled trial. So the virus trial that is done by Unicure, they did direct injection, but it wasn't felt that it was ethical to do injection of sham. So they basically put people under anesthetic and did a burr hole. And then 24 months later would go back in with another needle if they were going to get. So there wasn't a real placebo control. In this study, we can do placebo control, and the individuals who are in the placebo group can go on to get effective therapy afterwards. So I'm just at an hour, or it's 50 minutes into the talk, so I'm going to wrap it up. But this is our plan moving forward. We're currently engaged in these large animal studies in the Huntington's disease sheep. We think that this is going to validate and de-risk our route of delivery. It's the same Renishaw system that we'll use in people. We can see the potential benefit while the animals are still alive on these biomarkers. Of course, animal studies allow us to determine the efficacy in the brain. So we'll be able to see editing of the DNA, decreasing levels of RNA and protein in the brain and in the brain regions we're targeting. We are planning to have our first FDA and MRHA, the UK-based regulatory agency, early in the fall, and then a pre-IND meeting before the end of the year. This will allow us to get through the IND process and into our first-in-man trial, which we're hoping will be initiated at the start of 2027. We're planning to go into sites in the UK, Canada, and Australia, sites that have all previously been involved in some of the studies I've told you about. Our site here at UBC will certainly be one of the key sites for that first-in-man trial. So this is my story from a crazy idea. Can we encapsulate RNP within lipid nanoparticles that hopefully in the very near future will be brought all the way to human clinical trial? Much of this work has been incisive genetics, but a lot of it has come from people in the lab. Terry Petkow and Jay Kulkarni, Austin and Pam are all on the original patent for the method of manufacturing, as well as Peter Cullis that led to incisive genetics. This is the current people in my group. I mentioned Peter and Leslie, who have been collaborators with some of the work I've shown. Austin and Pam, again, lead the work in sites, and I'd like to thank you for your attention and time, and happy to answer any questions.
Thank you very much. Are there any questions?
A very great talk. I'm very excited about this therapy. It's truly transformative. I have a question. Why does this LNP ribonucleoprotein complex have to deliver intracranially, not just intrathecally?
Yeah, so the blood-brain barrier is a really significant barrier. We have done a little bit of work with intrathecal delivery into the CSF. There is actually a CSF brain barrier. My understanding is the things larger than proteins larger than 70 kilodaltons don't penetrate the brain very well from CSF. It's something we're still looking at, and one of the things I haven't talked about is targeting LNPs. So in our bone marrow work, we've actually, that work is moving forward with a modified LNP that has a targeting moiety, basically an antibody to a cell surface receptor in hematopoietic stem cells. We have similar work with targeting moieties that might get either across the blood-brain barrier, such as transferrin, or if you put it into the CSF. In the small amount of work we've done, when we put it into CSF, we don't see a lot of penetration. We think the lipid nanoparticles are just too big. But certainly that would be an approach. We've done some work with focused ultrasound. I was really enthusiastic about getting through the blood-brain barrier. So with focused ultrasound, you administer microbubbles into the systemic circulation or even intra-arterially, and then use focused ultrasound to burst the bubbles or to cause them to expand. That opens up transiently the blood-brain barrier, and then you can infuse your therapeutic agent. So there has been some data showing that we can get good penetration into the brain using focused ultrasound and LNPs. And I think that's very promising, but it's still, I think, further from clinical development. So we're actively involved in trying to get things into the brain, either from the CSF or the peripheral circulation, whether using a targeting moiety or using a focused ultrasound. There's certainly big advantages there. And I think that getting through the blood-brain barrier actually ultimately will give you the best full brain penetration, but those are simply further away from being able to move into patients right away. The system we're using is available. It's already being used in a number of human clinical trials. The Renishaw system has been primarily used to deliver growth factors in studies of Parkinson's disease. So yeah, I don't think that this approach is the end game, but I think it's the fastest way we can get this therapy into people. And again, I think first step is, can we get it to people? Can we show that it has benefit? We'll continue to improve how we do that in the future. So a great question, but I wouldn't say that intrathecal delivery is out of the picture. It's just, if you put in the currently available, clinically safe and efficacious LNPs, they don't get into the brain from the CSF efficiently. Hey, Phil.
Great talk.
Thanks.
Just two questions. So first, that delta H that created to include span right?
So we did. We have a number of different DNA templates, single standard DNA templates that we include. It's really interesting, actually. We actually got our best results with a DNA template that had nothing to do with the region. And in fact, I probably shouldn't be saying too much about this, but including a DNA template actually it has a greater effect on our encapsulation process. So we've got sort of a standard DNA template that we call it a editing enhancer. So it works with or without a template. Template improves our efficiency, but not for the reasons we thought.
So it's just small, because I thought it was really cool the way you took a snip and made it more selective.
Yeah, I didn't think that. I have to say, these are things that people in the I could give credit to the people who came up with it. I didn't think it would work.
So in theory, you take 2 steps instead
Mm.
and those to selectively target people.
I think you'd have a highly selective approach. There isn't we didn't have a SNP on the upstream side of the CAG repeat that worked well for us. So we did what we thought was, and again, full credit to the team, because this was not my idea. They said, well, if it cuts efficiently but doesn't affect transcription, then who cares? And so they found regions where small indels and cuts didn't really affect downstream. And so we thought that was a safe spot to have the first cut. And then but I completely agree, for other approaches, if you had 2 SNPs, that would be even better. That we tried that, we just couldn't find one that worked upstream of the CAG.
It's an apple of pain comment.
I absolutely think it should be, yeah. And you know, we built the platform, and now we're using the disease that I know best and we know best, where we have the resources and tools to bring it. But it certainly is a platform that can be applied to other approaches for sure. And our sickle cell work, I think, is probably potentially actually could be moving to the clinic as quickly as our Huntington's, but that'll be done with a partner who's skilled in that space, I'll say.
Hi, a fantastic presentation. Can you briefly repeat like which tissues can be targeted with your technology? I think you mentioned the liver, the eye.
Yeah. So
lungs.
when we established our company, one of the things that we were pushed to do was to build out the platform, and we did that for a couple of years. But it's by no means I would say exclusive. So there are other reasons. But we've been able to show really beautiful targeting to the cornea. So it's an area that is amenable to direct injections. So direct injections in the cornea, we got beautiful genome editing there, specifically in the stromal cells of the cornea. Systemically, it goes to liver very efficiently. Everyone knows that. Also, some of it does go to bone marrow and to spleen. So we haven't actually done systemic delivery of our targeted LNPs. So that's currently underway. And it may be that we don't even need to do intraosseous direct delivery, which is what we're currently doing. We may be able to do systemic delivery and get it to the bone marrow using our targeted LNPs. Direct brain. So really that's what we've done. There's a lot of interest in muscle, but I don't know whether muscle will work for LNPs. We haven't done it. So there's lots of tissues we haven't targeted. We haven't had success with everything. We really wanted to target retina, and we've done subretinal and intravitreal infusions, and we don't see good transduction or transfection in retina, and we're not quite sure why. It could be something to do with the specific region. So we haven't always seen good delivery, but those are the ones where we have tried and have seen good delivery. But there's lots we haven't looked at. We haven't extensively looked at spinal cord, which is absolutely an area of interest. And we think that intrathecal delivery might give us good spinal cord uptake, but we just haven't had the time to look at it. We do have a good system, and I haven't presented any of the data. So we use the TD tomato mice. So these are mice that have a red fluorescent protein behind a series of stop codons. And we actually have really beautiful guides that allow us to edit out the stop codons, and every edited cell turns red. And it's beautiful for those types of biodistribution studies. So if you have a tissue of interest and you want to test it out, we're happy to collaborate on that.
I was wondering what
It's a really good question, because we had thought early on that our straightforward, just we're going to lower Huntington was going to do it. And we had moved into the monkey studies because we thought we might be advancing that approach. At the same time, I mentioned the Roche studies in great detail, but other studies had failed. And there was this question that total Huntington lowering probably wasn't the right approach. So we actually, as a company, stepped back, because the technology was in the company at that point, and said, this is genome editing. We don't want to move forward with it until we're absolutely sure that we're ready. And so I actually set out a series of targets for the team, and we had a step back, and I said, I want to target everybody. I want it to be allele specific. I want to eliminate exon 1. I want to eliminate the possibility for somatic instability. I want to decrease mutant Huntington, and gave the team this target. So they went back and they said, okay, here's our approach. And I said, well, they'd already shown that they could do some large deletions, but they met the challenge. And so basically our approach was, what do you need to have the best in class therapeutic? Here's the guides, here's the targets. And they were able to check that this approach did it and show that it was efficacious. Then we wanted to show it was efficacious in human iPSC-derived neurons. And that actually is sufficient. We could go forward with a clinical trial with that, with our biodistribution data from non-human primate. But we want to further de-risk this by showing in the sheep. So really, that's what we need. We need the proof in the human cells and the sheep. And to be fair, they didn't hit every one of my targets. I wanted something that treats everyone. Our target, because we're targeting a SNP, only targets about 45% of the patients with HD. So they didn't hit everything, but they hit enough of them for us to hit the go and bring it into the path to the clinic.
And are there certain toxicity levels that are sort of required?
Yeah, I glossed over some of that. There was sort of a GLP tox studies. There are standard toxicity studies in rodents and in a second model, which we'll use the sheep for, and sheep or non-human primates are acceptable, as are dogs. So you really need 2 species and you need a standard set of toxicity. We've got some initial toxicity studies in rodents that look very promising, but that is usually done by a CRO. They basically have their standard techniques. You give them and at that point, you should really be giving them the GLP quantities. That's why I talked a little bit about manufacturing, because to get that final approval and IND approval, you don't have to, but you should have final clinical grade product that is in all the testing. So manufacturing is something we don't think about as scientists. It's critical for drug development.
I think we're out of time. Blair, thank you very much for talking to me.
Great question. Thanks.